Mitosis by Ethan and Max. The first stage of the cell cycle is interphase. In interphase, there are 3 steps, and the first step is the first gap phase. In this step, the cell grows larger and duplicates the cytoplasmic components in preparation for the second cell. The next step is synthesis. In this step, DNA replication occurs, and the chromosomes are duplicated for future duplicated cells. Finally, the cell goes through a second gap period in which the cell continues to grow and moves towards mitosis.
Prophase. Prophase is the first stage of mitosis. In this stage, the duplicated chromosomes condense. Next, the spindle fibers form on the centrioles, and the centrioles move towards opposite ends of the spindles. Finally, the nuclear envelope begins to disintegrate.
Metaphase. By this phase, the nuclear envelope has completely broken down and is no longer present. This allows the spindle fibers to attach to the sister chromatids and at the centromere, or the center of the combined chromosomes. Finally, the chromosomes begin to become further condensed and move towards the center of the cell, where they're lined up on the spindle equator.
Anaphase. During anaphase, the duplicated centromeres of each pair of sister chromatids separate, which result in the dotted chromosomes moving towards the poles, pulling away from each other.
Telophase. During telophase, the chromosomes decondense, which allows in two new nuclear membranes. Each membrane has its own cluster of chromosomes, which are identical in number.
Cytokinesis. Cytokinesis is the final process in cell division, but it's only possible after the completion of mitosis. In our case, our cell is circular and is an animal cell, so it goes through cleavage. A daughter cell is basically pinched off, leaving 2 separate